Today I'm going to talk about the admixture mapping that can identify the causal genes for lupus. So at the outset, I would like to say a few things about the genetic epidemiology of SLE and why I did the admixture mapping. SLE, as you know, is a very complex, common complex autoimmune disease. It is a strong genetic background, although it doesn't segregate as simple Mendelian diseases. So there are so many genes right now unknown, like some HLA and some non-HLA genes. And from our lab, we have discovered a few of the genes earlier. Although there are many genes unknown for SLE, but the functional variants, which is the actual causal variants, are not known for many, many genes. So as we have seen a few other earlier talk also, that African Americans has about 3 to 5 times increased risk for getting lupus. And so we thought maybe the ethnicity or the ancestry can explain the increased risk. So that's why we proposed the admixture mapping, which is a new mapping, gene mapping technique, which can be applied to the admixed population like African Americans. So what is admixture mapping? So think about the two ancestral populations like Europeans and Africans, maybe 200, 300 years ago, they come close together, and each generation there are some admixtures. And this is the current day African American populations. The idea of the admixture mapping is very simple. The disease-causing locus, near the disease-causing locus, if you look at their DNA, then most of the SLE patients who has shared that particular DNA will come from a particular ancestral group. So we are searching, that means we are searching the genomic regions which are associated with the increased risk, either is coming from the African ancestry or the European ancestry. So the measuring unit for this signal or the admixture signal is called the Lord score or the p-values. So in this study, this is the study design. So what we did about 1,000 cases. We did the entire genome scan. It's called the genome-wide admixture scan. And then we identify the signal, and then we follow the signal to localize any particular genes. And then when we localize their gene, we went there and identified the functional variants or the disease-causing variants, and then we replicated in African Americans and European Americans. And then experimentally, we also showed those variants, whether they are really causal or not. So we used about 20,000 SLE cases and controls. So when you search the entire genome, so this is one of these region of the genome, is chromosome 2. 2q22 to 24. It is about 21 megabase region. So in that region, we have the maximum signal, or the highest signal we got. The Lord score is about 6.3, which means it's a logarithm of the odds, which means that it is about 1 million to 1 odds that you have a signal that is kind of odds ratio. And that signal is giving that risk that you are inheriting the European ancestry. That means African American populations inheriting the genome from the European ancestral background, and that increases the risk about 1.5. That is 50% increased risk. So this region, this is the best region. So we followed that region about 19 genes with about 283 SNPs or single nucleotide polymorphisms. And as we said earlier, that about 20,000 samples from both African Americans and European Americans. When you search the region and you localize the gene, it's called IFIH1. It's also known as the MDA5. And that turns out to be 3 variants. Those are independently associated with lupus. And these 3 variants are located in intron 2, and 2 are exonic, or the coding variants. And this is the cases and controls from the African American. This is the cases and controls from the European Americans. And you can see the p-values, very, very significant p-values, 10 to the minus 15. Another is 10 to the minus 7. This one is exon 7 is 10 to the minus 7 for the African Americans. But this variance, particular variance, is very, very rare in the European populations. And if you notice, you can see that all three SNPs has A alleles are the risk alleles, and G alleles are the normal alleles. So where these SNPs are located, so this is the mRNA or the proteins of that of the gene IFIH1. The first one is the intron 1, which is located in the CART domain, and the second one is the exon 7, and which is located at the helicase ATP binding domain, and third one is located in the exon 15, which is in the rig-like 1 domain. So what is MDA5 or IFIH1? It is IFIH1 is a member of the RLR, rig-like receptor families. And IFIH1, main function of the IFIH1 is it senses the viral RNA or double-stranded RNA from the cytosol and initiates the antiviral responses by inducing different types of interferons and the expression of pro-inflammatory cytokines. Okay? So these are the three SNPs that we discovered by the admixture mapping or the statistical analysis. Then we are trying to see these are the three SNPs are located in 2 exonic and 1 intronic. So how those are statistically associated. Now the question is how they are, we can say that they are functional. So we did functional experiments. For the exonic SNPs or the coding SNPs, we did the apoptosis assay as well as this expression assays in the transfected cell lines. And for the intronic SNPs, we did the EMSA or electrophoresis mobility shift assay or the protein binding assay, and then we identified the bounded protein, and we also did some luciferase activity, luciferase assays to measure the activities of that particular SNPs, whether they're increasing or decreasing as a transcription factors. So in the transfected cell lines, we can see when we have this exon 7 SNPs, that time it is about 15% higher, increasing the apoptosis compared to the other SNP that we discovered. Then we are trying to see, okay, what are the different genes that is regulated by or that is connected with the IFIH1, and that if those 2 SNPs are functional, then this expression of these genes should be changing also. So what we did here, we are trying to measure the 11 genes. Those are associated with or somewhere connected with this IFIH1. We can see based on different types of the position of the exons, it is sometimes it is increasing some genes are decreasing. It has a nice pattern. And you can see that exon 7 changes the expression of NF-kappa B1 or CASP8 or CASP9 or MAPS, whereas exon 5 it changes the expression of NF-kappa B2 or other genes. And then it doesn't change anything for the one particular gene, that is REL-A. So based on these functions, we can tell the expression of the other genes depending on where you have the risk allele in the exon 7 or exon 15. So depending on that, we can tell that this is a functional, these are the 2 functional SNPs. How about the other SNP that is located not in the coding region, but located in the intronic region? So as we said, we had done the EMSA, electrophoresis mobility shift assays, and then we had identified which protein it binds. And based on that, you can see the EMSA assay is shifted, and when we identify these proteins, and it looks like, and we also sequence that proteins, and it looks like the 2 proteins mainly it's binding there. One is NCL or nucleolin, and other is KU70, KU80. And then we ask the question whether this particular SNP in that region, whether it can use as an enhancer, because it is changing the expression. So we did the luciferase assay, and based on this, we can see that when you have the G allele, at least it increases 2-fold increased risk. But on the other hand, when you have the A allele, the risk allele, it decreases the expression. So based on that, we can tell that this is also a significant SNP, and it is also a functional SNP. So these are the 3 functional SNPs we identified from here. And we proposed, from these 3 functional SNPs, we proposed a functional model, how it works. And it looks a little bit complicated, but looks like the 3 SNPs in these regions located, and they act independently and ultimately causing the inflammation and causing the may be causing the lupus. So the whole work, I'm very happy that it just published this last month in PLOS Genetics. So we're very happy about that one. And at the end, I'd say like to acknowledge to my group. And as you say, it's a lot of 20,000 samples, a lot of collaborators actively working together, and funding from the NIH. Thank you very much.